Everybody. Thanks for coming to my talk, and I'm looking forward to telling you about the work we've done over the last year and a half rearing the endangered sunflower star Pycnopodia helianthoides. So the purpose of the project is to explore the feasibility of a captive rearing program for the species. And we also would like to understand key aspects of their life cycle, in particular, poorly understood settlement and post-settlement juvenile stages, in order to gain insight into the ecological function of Pycnopodia helianthoides, the sunflower star, through its ontogeny. So a little bit of background on Pycnopodia declines. You've probably already heard in this session about sea star wasting disease. It's impacted every known species of sea star in the Northeast Pacific, and it peaked about 5 years ago. And of all the species that were affected, sunflower star, Pycnopodia helianthoides, a major predator, was seemingly the most severely impacted. And the loss of this predator in California has been associated with explosions in the populations of one of its prey, purple urchins, and then associated declines in kelp. So they all may be related. There are remnant populations of sunflower stars in the state of the sea, thankfully, and that includes the San Juan Archipelago where we did our work. So a quick overview. I'm going to tell you about working with adults. Then we're going to go over reproduction, and then fertilization and larvae, settlement of larvae to the seafloor, juvenile culturing, and I won't really talk about transport of different life stages, but we have done it successfully, sending gonad in the mail, transporting individuals between locations in Washington. And so I'm happy to fill anybody in on the techniques that we used if you're interested. So the adults, populations of the sunflower star and the San Juans are present both intertidally and subtidally, but not in all the places that we used to find them. We do see some relative hotspots though, and using that information, we've been able to collect 30 adults now. And we had some examples of wasting initially, but in the last 16 months, we've had all 30 of those adults with no wasting. We've been feeding them mussels every other day, and we've been measuring them too. They've grown at a rate of about 17 centimeters per year over that period. We can also ID individuals, and Alexi is going to tell you about that. You can see a little bit of that color variation below. Incidentally, those small juvenile stars are fun to take pictures of. We don't collect those for our breeding colony. We return them to the field, but I wanted to show you some photos of some juveniles we found. So reproduction, we monitor reproduction by hormone injections, amputations, looking at dropped arms when we did see wasting, and one example of spontaneous spawning. Briefly, the hormone injections don't work very well. We think that may be due to their relatively low reproductive output. So instead we've used amputations, which has worked really well. The only time that we've seen large female gonads is in animals that were wasting, which we think may be related, but we don't really know what the mechanism of that might be. The reproductive season, as we've seen it, looks like it's November to April. And in fact, we're just getting ready to do another spawning next month. Or this coming month by the time you all hear this. And so that seasonality, November to April, is pretty much the opposite of what it says in the literature. Successful fertilizations, we were able to accomplish them using gonad shipped to us from Alaska, as well as gonad that we recovered from amputations in November and April of last year and April of this year in Washington. And we've seen spontaneous spawning once in May. And so that's why we think that November to April is probably the normal reproductive time, and occasionally they then spawn out if they're held in May. After spawning, the eggs are fertilizable for about 1 to 2 days at ambient temperature, the sperm for about a week if you keep them in the fridge, and we're currently testing whether sperm can be cryopreserved at deep freeze. So here's some photos. Upper left there is our sea star Olga, this is the male, and he is covering over his wound here. That is not arm twisting behavior that you might be familiar with as a result of wasting. This is normal arm twisting behavior that they do when they're covering over a wound. And as I said, they recover really well from this. We recovered gonad, tends to be pretty small. Here's an example of gonad from a female, a different individual than Olga. And you can see it's quite a little piece there, but it was enough to be able to get eggs out and start a larval culture. In the upper right, that's actually the arm of a sea star from Hood Canal that wasted at the Seattle Aquarium, and you can see that it's got a pretty big gonad, relatively speaking. And so that's related to what I was talking about wasting. In the lower right, that's a male spawning, and you can see the sperm coming out of the gonopores there. And so fertilization's in larvae. After you dissect the gonad, the eggs will complete meiosis. If you put them in that same hormone 1-methyladenine at more dilute concentrations than you would inject. Fertilized eggs have a fertilization membrane, and they're actually much larger than the literature suggests. That's another difference with the literature. They're 160 microns in diameter, both the eggs from Alaska and Washington. That's 40 microns in diameter bigger than the literature says. Larvae grow well with standard methods. We feed them a combination of two microalgae, Denaliella and Rhodomonas. They begin to form their rudiments at about 25 days post-fertilization. That's the beginning of the process of metamorphosis, which then takes, their juvenile rudiments grow inside the larva. The metamorphosis happens while it's still a larva, and then it settles to the seafloor, as I'll tell you, starting at about 50 days. The ontogeny is similar in Alaska and Washington. The skeletal elements that form in the rudiment are useful for staging, as well as some other features I'll show you in a second. Here on the upper left is a gonad that we treated with hormone, and you can see it spilled all its eggs out after about an hour. When you do a close-up of those eggs, you can see that they've got these little clear areas. Those are the germinal vesicles. These are immature eggs that are not ready to be fertilized, but you leave them in hormone for about an hour and those germinal vesicles break down. You got nice opaque eggs without that clear spot. And this is a close-up on the lower right. Those are now ready to be fertilized with a dilute suspension of sperm. Here's some pictures of ontogeny during larvae, during embryo and larval development. There's a 2-cell stage in the upper left and a gastrula below that. Lower left is an early stage larva. Here's a typical bipinnaria at 16 days. At 24 days, it's becoming a mature bipinnaria, and it's getting ready to start forming its juvenile structures up here. And here at 40 days, you see a brachylaria larva, and this halo right here is what we call the helmet, which suggests to us that it's competent to settle when it's got that structure, that skeleton that you can see, that white part. Here are some close-ups of that, those skeletal structures and the things we use for staging. Here's a brachylaria larva under cross-polarized light. This glowing stuff is its calcium carbonate skeleton. Below that is a close-up of that, looking at some of the skeletal elements, quite beautiful. And to the lower middle there is a helmet stage. You can see the skeletal elements within the helmet. Up above that here, you can see the 5 little bumps. Those are the start of the primary podia, the 5 podia of the 5 arms, basically, of the incipient sea star within the larva here. And here are some structures in the brachiolar arm that we also use for staging. We obtain the first settled larvae at 50 days, peak at 60 to 70 days. They settle at about 0.5 millimeter in diameter. Due to larval cloning and other factors, there's asynchrony. Many larvae were delayed, and some settled even 4 months after fertilization, and we released some larvae at that point. So they can persist for a really, really long time in the plankton. Larvae will settle spontaneously in jars, and they adhere so strongly that this can be a problem with respect to collecting the juveniles because it's hard to get them out of the jars. If you thoroughly clean the walls of the larval culture vessels every 2 days at the end of larval development, it reduces this spontaneous settlement. Larvae also show a strong specific settlement response to biofilm grown in the presence of adults. They will also respond to other biofilms, but much less readily. They respond to the coralline alga, Calliarthron tuberculosum, and settle. And they'll settle directly on the fronds, as I'll show you. And turbulence exposure heightens the response of their larvae to the Pycnopodia biofilm. And Florento, tomorrow at 1 p.m. will be telling you about that. Here's some settlement data. You can see proportion settled after 48 hours in a queue of urchin biofilm, of Pycnopodia biofilm, and of Calliarthron. And here's an image of a bunch of little juveniles settled on a Calliarthron frond. And here are some images of settlement. This is a larva settling, attaching here with its super long posterolateral arms. This is it retracting. This is, we call this a settler. It hasn't quite settled. This, we call it settled. This is a little juvenile. And you can see the juveniles here are all gathered around a little piece of dead Calliarthron, which I thought was really interesting, and I don't know what it means. So we have attempted to culture the juveniles, and it took us a while. We lost a lot early on, 99% until we figured out what we were doing. We had several bouts of mortality due to overgrowth of fouling diatoms and ciliates and other reasons. Cannibalism is also a source of mortality, as Shannon will tell you tomorrow at 1:15 p.m. And juveniles do much better in flow with significant oxygenation and benefit from micron filtered seawater and the presence of Calliarthron in their culture vessels, from what we can tell. It's not clear exactly when the juveniles start to feed and what they eat in the initial weeks, which we think is a critical period. They're likely grazing during that time. Then they start to cannibalize one another if they're really dense. And they'll also eat newly settled echinoid juveniles, which we grow for other reasons, and we're really interested in that ecologically as well. And so that makes it a good food source in those early stages and to keep them from cannibalizing one another. When they reach about 1 millimeter, we've been feeding them crushed juvenile bivalves that we got from Taylor shellfish, manila clams, and Pacific oysters that are smaller or about the same size as the juveniles. And at about 1 centimeter diameter, they'll actually actively feed and open those juvenile bivalves. They start with 5 arms, they add a 6th at about 5 millimeters, and they can add new arms at least 3 different ways. I'm not going to show you that. Here's a culture vessel completely occluded with that benthic diatom that kills the juveniles. Here's a bunch of cute little recently settled juveniles. You can see their arms grow. They start to add additional arms, and this is the best culturing condition we have, recirculating seawater in a temperature controlled chamber with some water flow and oxygenation. We had a lot of mortality. I won't walk through this in great detail, but you can see that in various different culture vessels cultured at Friday Harbor or aquarium in natural seawater did really poorly. We've had better success more recently with recirculating seawater in the conditions I showed you. And the rate of death has really slowed, and now they're basically surviving, which is great, even though we don't have very many left. And this is the growth rate of the juveniles. And in the interest of time, I'm going to move on. But these are our champions that are almost 2 millimeters now. And you can see that it takes some quite a while to start to get going. And once they do, they kind of seem to ramp up in their speed. So these early stages down here seem to be the most critical. And lots of people to thank, not enough time to thank them all. These are all stars of the project. I want to thank especially the Nature Conservancy for funding. And thanks so much, everybody on this slide, and for all of you for your attention. And enjoy the meeting.